For the first part, we have introduction. Environmental analysis is the process of collecting and analyzing samples from the environment, such as air, water, and soil, to evaluate their quality and detect the presence of pollutants or contaminants. It plays an important role in protecting human health, preserving ecosystem, and ensuring compliance with environmental regulation. Among the various fields of environmental analysis, our group will focus on water analysis, as maintaining good water quality is essential for drinking, agriculture, industry, and aquatic life. One of the most widely used techniques for water analysis is spectrophotometry, which measures the amount of light absorbed by a substance at a specific wavelength to determine the concentration of pollutants such as nitrate, phosphate, and ammonia. Due to its accuracy, reliability, and rapid analysis, spectrophotometry has become an essential tool for monitoring water quality and supporting environmental protection efforts.
Knowing nitrate levels is important because they affect both the environment and human health. Excess nitrate in water can cause eutrophication, where algae grow rapidly and reduce oxygen levels, harming aquatic life. For humans, drinking water with high nitrate levels can cause methemoglobinemia or blue baby syndrome, which reduces the blood's ability to carry oxygen, especially in infants. So the main objective of this experiment is to determine the concentration of nitrate nitrogen in 3 different water samples using a UV visible spectrophotometer. In environmental analysis, spectrophotometry is commonly used to determine the concentration of substance in water samples through the colorimetric method. Many pollutants in water are colorless and cannot be detected directly. Therefore, a color-developing reagent is added to the sample which reacts with the target substance to produce a color compound. In this experiment, tap water, bottled water, and river water are treated with the same color-developing reagent under identical conditions. Comparing their absorbance values allows the concentration of the target substance to be evaluated and the water quality of each sample to be compared.
I will continue to methodology. For the first methodology is personal protective equipment. Personal protective equipment including lab coat, goggles, latex gloves, long pants, shoe, and medical mask. We need to use this in lab for protecting ourselves from chemical. Next, I will talk about water collection and filtration. Water samples were collected from 3 different sources: commercial water, tap water, and river water or pond water. Each sample was stored in clean glass container and clearly labeled. The samples were then filtered using 0.45 micrometer membrane syringe filter to remove suspended particles and reduce turbidity, which could interfere with the spectrophotometric analysis. The first 1 mm of filtrate was discarded to rinse the filter membrane before collecting the filtered samples. Next, I will continue to preparation of calibration standard. 6 standard nitrate solution with concentration ranging from 0 to 5 mg per liter were prepared by pipetting different volume of the working nitration solution and diluting each to approximately 50 ml with distilled water. At the same time, 50 ml of each filtered water sample were transferred into the separate evaporating dishes. Next is evaporation and nitration. All standard and sample were evaporated to dryness using hot water bath. Once cool, 2 ml of PDA reagent was added to each dish. A clean glass rod was used to spread the reagent evenly so that the dry residue dissolved completely. Finally, the dishes were left undisturbed for 10 to 15 minutes to allow the nitration reaction to occur before proceeding to next color development and spectrophotometric measurement.
Next, onto the color development and volumetric makeup. First, carefully add about 20 ml of distilled water to each dish to dilute the acid. Next, working in a fume hood, slowly add liquid ammonia drop by drop to each dish while stirring gently until the solution becomes alkaline. Next, quantitatively transfer the yellow solution from each dish into a separate clearly labelled 50 ml volumetric flask. And then rinse the dish twice with a small portion of distilled water and add the rinsings into the flask. And lastly, dilute each flask exactly to the 50 ml calibration mark with distilled water. Stopper the flask and invert them several times to mix them thoroughly. Now, onto the spectrophotometric measurement. First, switch on the spectrophotometer and allow it to stabilize for 15 minutes. Set the monochromatic wavelength to 410 nanometers. Next, rinse and clean the cuvette with the bank solution. Fill it, place it in the chamber and adjust the instrument to read about 0.00 absorbance. Next, sequentially measure and record the absorbance value of the remaining standard solution. Lastly, measure and record the absorbance value for sample S1, S2 and S3.
Representation of data. As you can see here, the table 1 shows the outline of calibration curve data. The graph here is plotted based on table 1's data. Using the standard formula for a straight line, y equal mx plus c, where the y-intercept c is negligible or false to 0, we get y equal 0.113x, where y is the absorbance value read from the instrument, m is the slope of the line, and x is the nitrate concentration. Calculation and analysis. Here we have the calculated nitrate concentration x using the formula y over m. For the bottled water, we get 0.15 mg per liter, and for tap water we get 1.30 mg per liter, and for river water we get 6.50 mg per liter. Based on this calculation, we did an analysis. For the bottled water, it is highly pure and pristine baseline condition, and for tap water, it is safe, well within the standard of EPA, WHO, drinking threshold of 10 mg per liter. And for river water, it is elevated risk, approach the critical ecological limits, and have 2 points to agriculture runoff or local sewage contamination. Interpretation of result. As the concentration of nitrate increases, the measured absorbance increases linearly. This perfectly demonstrates Beer-Lambert's law, which states that absorbance is directly proportional to concentration. Water samples with higher nitrate concentration indicated a greater level of contamination. The results were used to assess the suitability of the water for environmental or domestic use.
So, the experiment has successfully fulfilled the objective by determining the distinct nitrate-nitrogen gradient across 3 water sources, which is bottled water, tap water and river water. The bottled water and tap water sample both fall safely below the strict 4.5 mg per liter maximum permissible limit by the Bureau of Indian Standard and the World Health Organization for drinking water. This confirms that the commercial reverse osmosis and the municipal water treatment plants are highly effective at ensuring public safety against the nitrate induction risks like methanoglobomania. Conversely, the river sample exhibit a significant elevated nitrate level, which is still technically below human drinking toxicity limit. This level points a localized nitrate pollution from agriculture, fertilizer runoff, domestic wastewater, and the natural nitrate waste from organic matter. To In an open aquatic ecosystem, nitrate level exceeds 1 to 5 mg per liter poses an ecological threat by triggering an eutrophication. So we can conclude that this experiment demonstrates that spectrophotometric analysis of 410 nanometer are reliable tool for environmental water monitoring and highlighting the exceptional purity that the drinking water supplies, while proving the critical need for watershed management and to protect the habitat.